Hello, Kevin. How you guys doing? I'm mucking with the lights. It's hard to get the screen the way I want it to look on the camera, and I discovered that there's a light that just blasts at it right there. So hopefully this will help that situation. How's everybody doing? You guys watching the videos?
Yes.
Good? Anybody having any troubles with the videos? Anybody having any trouble understanding the material? Yep. Nobody will say yeah to that, you know? Yeah? There's one honest person back here? Yesterday went a little fast, yeah. I covered quite a bit of territory yesterday. Yeah, I did that. So it's always hard from my perspective realizing that, and sometimes it's after the fact, and I'm going to go, oh, wow, we don't have that much more we have to cover tomorrow. So that's when I usually realize that I've been going pretty whirlwind. So things making sense?
Yeah.
Good. You guys work through problems and things like that?
Yeah.
Book helpful or not?
Yeah.
Okay, good. What's that?
Do you take your own pictures?
Yeah, I take all my own pictures, yeah. Good. I decided with the book that since it's not being printed, that there's no limit on length. So I could take and make what I described as I want to make the most beautiful appendix anybody had ever put into a book. So that's why all the limericks and all the pictures and all those things are there. So I hope you enjoy those. Okay. We're on a very interesting topic, and protein structure may seem rather arcane. I hope yesterday you started to see something about that. I had a very neat experience after yesterday's lecture. I had an email from, or actually a YouTube message from somebody watching the videos at the University of Houston. And they watched yesterday's video, and they said, I can't wait to hear the hemoglobin lecture, because I said yesterday it was one of the most popular lectures. So if you're watching in Houston, I hope you enjoy this. All right. So all that silliness aside, let's get to the topic of more dimensions of protein structure. So yesterday I got a reasonable start talking about tertiary structure. So I hope that what I communicated to you was that tertiary structure is very important for proteins, because tertiary structure involves folding. We saw how those bends and those turns and all those things, all those different components arrange themselves in three-dimensional space. And though I've talked about it a little bit, I hope that you'll start to see that structure is absolutely essential. I talked yesterday about cooking an egg. I talked about how we kill bacteria by heating them up, because we denature or unfold their proteins, and with the unfolded proteins they don't function, and the bacteria or any cell will die with that. Okay? I'm going to elaborate on that a little bit more today, and when I start talking about hemoglobin, I think and hope that you will start to understand how even tiny changes in the structure of a protein can have some enormous effects. Okay? All right, so that's part of what I'll be talking about. Now, you'll notice I said I was going to talk about phi and psi today, and I'm actually going to save that, and I might not talk about phi and psi at all, in which case we'll have one less thing to know. All right, what you see on the screen is an oxygen-binding protein called myoglobin. And myoglobin sounds like hemoglobin, and with good reason, because they both are part of a family of proteins known as the globins. All right? And myoglobin is very closely related to hemoglobin. One of the differences with myoglobin is it only has one protein subunit. We'll see that hemoglobin actually has 4. So it only has one protein subunit. And the structure of that one protein subunit is not unlike the structure of each of the 4 protein subunits that we see in hemoglobin. Like hemoglobin, myoglobin binds to oxygen. But myoglobin doesn't function to transport oxygen. Instead, myoglobin functions as what I call an oxygen battery. All right? What is an oxygen battery? Well, if we think about a battery that's in my laptop computer, if I unplug my computer from the wall, the battery kicks in and nothing happens, right? I don't lose anything. I've got all the stuff that's there, right? It's a battery backup. An oxygen battery backup is something that myoglobin provides to your muscles. It turns out that you can exercise so fast that you use oxygen faster than your blood supply can deliver it? Well, if you start running out of oxygen, okay, believe it or not, you don't die immediately. You have an ability to keep things going, even when you're completely devoid of oxygen in your muscles, that is. But for a short period of time, myoglobin can give up some oxygen and let the muscles have its oxygen. So it's like an oxygen battery. We'll see when we talk about hemoglobin that the most important component of that oxygen battery is right there. Okay? That's the part that gives hemoglobin its name. It's called the heme, H-E-M-E. And myoglobin has a heme. We don't call it hemoglobin, but it has a heme also. And the heme contains an iron atom right in the middle of it. And that iron atom is what binds to the oxygen. Now, what we're going to see is that even though myoglobin has a structure of one of its only subunit, very much like the four subunits in hemoglobin, myoglobin and hemoglobin have very different properties with respect to oxygen. Okay? Now, one last thing I'll say about this structure as we look at it is that if we look at this, we don't see any beta strands in here at all. Everything in myoglobin and everything we see in hemoglobin is alpha helix. Alpha helix, turn, alpha helix, turn, alpha helix, turn, alpha helix, turn, etc. So we see the arrangement of these various alpha helices in three-dimensional space are a function of the angles of those turns. Okay? Very, very important component. Yeah. So the last sentence was that we see that the structure of myoglobin in three-dimensional space is a function of the angles of each of those turns. Okay. So the turns play a really big role in determining that overall structure of a protein. All right. Now, I'm briefly going to just mention to you a couple things that we do. How do we know the structure of a protein? Right? So I just want to say a couple words about that. This is not a biochemistry class. We're going to learn how to draw that structure or anything like that. But I want to say a word or two about that. Right? Determining the structure of a protein is something that biophysicists do. I'm in a department of biochemistry and biophysics. Okay? And people always say, well, what is biophysics all about? So biophysics is about determining the relationship between structure and function. Biophysicists are the people who determine those protein structures. Okay? Now, I always like to tell students that are visiting when they're asking about what biophysics is and so forth, that a biophysicist uses techniques called spectroscopy. Spectroscopy can involve basically electromagnetic radiation that a biophysicist basically exposes a molecule to. And how that molecule reacts or interacts with that electromagnetic radiation tells a biophysicist what its structure is. And that structural information can be very, very detailed. A biophysicist using techniques of spectroscopy, and yes, X-ray crystallography is one, and nuclear magnetic resonance, that's NMR, is another one. A biophysicist can use these two techniques and determine the position in three-dimensional space. So we have three dimensions out here. In three-dimensional space, they can determine the position of every atom in a protein that might have 20,000 atoms. Well, A, that's a lot of information. B, that's a lot of atoms. But C, that's really useful stuff. Because if this protein is, for example, an enzyme, this enzyme catalyzes a reaction. If I know where all the atoms in the enzyme are, I know the shape of the molecules that will fit into that enzyme. I know the location of where the reaction will be catalyzed. And if I'm a drug designer, I also know how to design a drug to plug up that enzyme because I know the exact dimensions it's got to have. So knowledge of structure really allows us to do a lot of things. Biophysics is very, very useful for that. And that's why we do techniques called X-ray crystallography, which involves shooting X-rays through something we're trying to determine the structure of. Our nuclear magnetic resonance, which involves using electromagnetic radiation to do that. This building has a very big NMR in it. Okay? Very powerful magnet. Very, very powerful magnet to do stuff. Okay. So those are kind of cool techniques. I said that the most important component of myoglobin, from the oxygen binding perspective, was the heme. Okay? The heme is composed, this is actually the structure of the heme, and it comes from this molecule called protoporphyrin 9. That's the sort of foundation of heme. You put an iron in the center of there, and you have heme. You put a magnesium in the center of there, and you have chlorophyll. So we see that heme and chlorophyll are closely related. They have very different functions, but they are closely related. Okay? So when I talk about the heme group, this is the guy that I'm talking about in that center of that myoglobin or hemoglobin molecule. Okay? Question? Oh, okay.
The heme has an iron, but what does the chlorophyll have again?
Chlorophyll has a magnesium. Yeah. Was that your question back here? Yeah. Okay. All right. So we're going to focus on hemoglobin. Okay? And when we look at that heme group, what we see is something that looks like this. So we're looking very up close and personal inside of this myoglobin or hemoglobin. The same thing happens in both of them, where we have this iron atom and we have an oxygen molecule that wants to come in and bind. Okay? Now, when this oxygen molecule comes in and binds, it does something really interesting. Okay? If I think of this heme, I can think of it as a very flat thing, right? It's just a flat, planar structure. Well, in fact, it turns out it's not completely flat. Okay? It actually is slightly concave. So I'm exaggerating it with my hand, but the iron would be right here at the base of my hand. That's before the oxygen gets in. When the oxygen comes in, the oxygen pulls the iron up, and this thing becomes much more flat. Everybody understand that? So no oxygen here, and then oxygen comes in, and it pulls the iron up, and the whole thing flattens out. Now, that very, very tiny change in structure has enormous implications for you as a living animal. Without that little thing I just described to you there, animal life would not exist. Now, you don't know why yet. I'll tell you why. But without that very tiny structural change, animal life would not exist. So that's where we're headed. All right. Well, what else about this guy? Well, one of the bad things about this is that heme really is very capable of binding carbon monoxide. One of the reasons, there are actually two main reasons, but one of the reasons that carbon monoxide is poisonous is it can bind to hemoglobin instead of oxygen, and we starve our cells for oxygen. If you look at these two guys, they're almost identical in shape. Almost identical in shape. All right. So that's what's happening. So, back up one more thing. So, if I take this guy here, it's flat, and oxygen binds, and it makes it go up like that, I've moved the iron upwards, very slightly. Well, notice that the iron is connected to a histidine. That histidine is part of the globin part of the molecule. The histidine is attached to another amino acid, which is attached to another amino acid, which is attached to another amino acid. We've got a whole chain of amino acids, right? So if I pull up on the iron, I'm slightly changing the position of the histidine, which means I'm slightly changing the position of whatever's attached to the histidine, which means I'm slightly changing the position of whatever's attached to the histidine. You get the idea. The toe bone's connected to the foot bone, and the foot bone's connected to the ankle bone, and the ankle bone's connected to the shin bone, and if someone pulls my toe, my leg moves. Right? Okay. I want you to keep that image in your mind as I get talking about hemoglobin. Okay? It happens in both myoglobin and hemoglobin, but only in hemoglobin is it important. Before I talk about hemoglobin, your book has a little side, I shouldn't say your book, because this originally was focused on another book, so that's where my lecture came from originally. But another thing we need to consider is the structure of proteins and how we can change those structures. I talked yesterday about how we can take that egg, we heated it up, we denatured the protein. Well, denaturing simply means unfolding it. It simply means unfolding it. Okay? So what you see here is the unfolding. And what this is illustrating to you is something really important about the unfolding. Okay? The unfolding, this is an enzyme called ribonuclease. This enzyme really, really, really doesn't like to be unfolded. In fact, most enzymes, it's pretty easy to unfold them. We heat them up, they unfold, and everybody's done. But this guy has some very, very important disulfide bonds that really help to hold it together. Remember, the disulfide bonds are covalent bonds. Okay? And I said yesterday, if I try to heat, I cannot break a covalent bond with heat unless I go to like 1,000 degrees. So I can't break that. Ribonuclease, if I take it and I heat it up to boiling, ribonuclease, when it cools back down, will still be active, meaning it hasn't unfolded. Now what this tells us is that ribonuclease, first of all, resists that unfolding. If I want to unfold ribonuclease, I have to basically take its support system away. Right? And the support system are those disulfide bonds. If I want to effectively do that, I've got to break those disulfide bonds. Well, it turns out that this chemical right here, beta-mercaptoethanol, okay, breaks disulfide bonds. In this case, I'm also using urea. Urea is the stuff in your pee that stinks. Okay? Urea also has the property that it will break hydrogen bonds. So I can heat it up and break hydrogen bonds, or I can use urea to break hydrogen bonds. But in either case, if I do either of those with beta-mercaptoethanol, I will unfold ribonuclease and make it inactive. For most proteins, I don't have to do that. And you might conclude, oh, most proteins, therefore, don't have disulfide bonds. No. Most proteins do have disulfide bonds. But they don't have them in the critical places that help give structural integrity. So we can think of the disulfide bonds in ribonuclease as being like the support beams in this building. Pretty darn important. We take those support beams away, the building's not going to be very stable to stand up, and this protein's going to fall apart. If you think about other proteins that have very stable beams, very stable support beams, maybe those support beams aren't really important for their structure. So we can break those, we don't need to break those, and the structure still falls apart. It's kind of like being in a building like Waldo Hall where there's no great earthquake support, right? Waldo Hall in an earthquake is probably going to go down to dust. All right? Most proteins are like Waldo Hall. The support beams aren't really there in the same way that they are inside of ribonuclease. Does that make sense? Questions? Okay. Okay. Now, most proteins will not come back together. The egg, I said yesterday, we can't unscramble an egg. But it turns out I can unscramble ribonuclease. That's kind of surprising. Most proteins won't do that. Ribonuclease is a really unusual protein. Because if I'm really careful, I take it apart, and I allow things to happen, ribonuclease will come back in exactly the way it was to start. If I were to ask you on an exam what that meant, what would you interpret of that? What would that tell you? It can reform the disulfide bonds. My question is, what does it tell you about folding? But you're right. Okay, so you're on the right track. It's folding. You're saying it only fold one way. It turns out things will fold more than one way. So that's not completely true. But what's necessary for folding? Primary structure. The only thing that's there once we've unfolded is the primary structure is still there. In the case of ribonuclease, the primary structure is enough to drive the folding process. Why don't other proteins do that? Why don't other proteins do that? The reason other proteins don't do that is that there are enormous numbers of other possible folds, Ribonuclease's primary structure leads it most of the time to the proper fold. The others don't get there. Well, how do the others get folded properly in the first place? Okay, what's that? Ribosomes make the protein, okay? And the answer is part of what you're saying here, okay? Most protein folding starts as the protein is being made. So it doesn't have all those other distractions. I don't have one end interacting with another end, which is what I would have right here, right? Because as this is being made, this is only interacting with the very next thing, the next thing, the next thing, the next thing, the next thing. That's a very different thing than taking everything apart and saying, okay, now you go back together. Ribonuclease has the ability to do that. Most proteins don't have the ability to do that. But ribonuclease does tell us that there's enough information in sequence to direct the folding. That's a very important message we're getting from that. Okay, now let me get away from ribonuclease and come back and talk about hemoglobin. All right, hemoglobin brings up another level of protein structure. We've talked about primary, which was the sequence of amino acids. We talked about secondary, which was the interactions between amino acids that are close in primary sequence. We talked about tertiary, which were interactions between amino acids that were not close in primary sequence. And now we talk about quaternary. Quaternary relates to interactions between different polypeptide chains. All the ones we've talked about so far were within a single polypeptide chain. Now with quaternary, we have different polypeptide chains that are interacting with each other. Okay? Hemoglobin, as I noted yesterday, has 4 polypeptide chains. 2 of those units are identical. They're called beta. The other 2 are identical, and they're called alpha. So we describe the structure of adult hemoglobin as alpha 2, beta 2, because that's what comprises it. The alpha subunits are very much like the beta subunits in terms of structure and in terms of function. The alpha subunits and the beta subunits are both like globin, like I said before. Did you have a question?
Well, it has 2 chains or 4 chains?
It has 4 chains, 2 identical copies of 2 each.
Okay.
2 identical alphas, 2 identical betas. Okay? Now, it turns out that having 4 chains gives hemoglobin some properties that are very different from the properties of myoglobin. I'm going to tell you in words what it is, and I'm going to try to illustrate it for you also. Let's think about it in words. Let's think about that foot bone connected to the ankle bone, connected to the shin bone, connected to the knee bone, etc. Right? I've got 4 chains. Each one has a heme. There's a heme, there's a heme, there's a heme, and there's a heme. Each one has 1 heme. Each heme can bind 1 oxygen molecule. When hemoglobin is traveling through our body, it gets to the lungs, and it's already dropped off all of its oxygen having gone through the body, doing its job, dropping off oxygen. It gets to the lungs, and there is no oxygen in it. Okay? Hemoglobin's empty. That's good, because it's dropped off its oxygen where it was needed. It gets to the lungs, and the oxygen concentration in the lungs compared to the rest of the body is very high. It turns out that hemoglobin is in a state where it doesn't want to bind oxygen very much. It's called the T state. T, the letter T. Okay? Fortunately, it doesn't stay in that state very long, because what happens is one of these hemes encounters an oxygen and binds to it. Now what happens? We pull up that iron a slight distance. That iron pulls up a histidine a slight distance. We can imagine we're going to change the structure of one of those subunits very, very slightly. Right? Okay? Well, that one subunit is interacting. Let's say it's this subunit right here. That subunit is interacting with this subunit. It's interacting with this subunit. And it's interacting with this subunit. And guess what? Those interactions affect each of those subunits. Now, the structure of the enzyme changes from what's called the T state to something called the R state. R, the letter R. Okay? R stands for relaxed. T stands for tight. In the relaxed state, hemoglobin really likes to bind oxygen. So what's happened? We have converted hemoglobin from a state where it doesn't like to bind oxygen into a state where it really likes to bind oxygen. Well, that's really important because hemoglobin isn't in our lungs for very long. And during that short time it's there, it would really be nice for it to bind as much oxygen as it possibly could. So because of this one minor change that happens in this subunit, this one affects this one, which now binds oxygen, which affects this one, which affects this one, and all of a sudden hemoglobin gets loaded with oxygen. I said that was essential for life as animals. The reason it's essential for life as animals is animals have to run, they have to catch prey, they have to escape prey. That takes muscular energy, and muscular energy takes a lot of oxygen, and that's why we have this. Yes, sir?
Does oxygen preferably bind with either the alpha chain or the beta chain?
It doesn't preferably bind to either one. You can treat them independently as if they're the same thing. Yeah?
So what minor change in one of the subunits affects all of them?
Yeah. So, and it's sort of sequential. We can think of this one affecting this one, affecting this one, affecting this one. But yeah, in essence, you're right. Question there? Yeah. Go ahead. Yeah.
So does the T state happen in the lungs, or does it happen in the tissues because it doesn't have
Good question. By the time it's back at the lungs, it's in the T state. And I'll say a little bit more about that in a bit. But it's a very important point. Yes?
How do you get that first minor change to happen?
How do you get that first minor change to happen?
It's in the T
The first oxygen has got to bind. I didn't say it was impossible to bind. I just said it's not preferable. So one of the ways, it's a good question, one of the ways in which that happens is that the oxygen concentration is very high. Oxygen is forcing itself in there. After that first one goes on, the next ones go on really readily. The phenomenon I just described to you is called cooperativity. Cooperativity. That's where the binding of one molecule favors the binding of other molecules. The very first one favorably affects the other one. Was there a question back here? No? Okay. Cooperativity is very, very important. Okay? And it's all happening because of that very slight change from being concave to flattening out. That very tiny movement, which is a fraction of an angstrom of the iron atom, is changing hemoglobin enormously. Okay? Now, graphically, here's what it means. This shows the binding curves for myoglobin compared to hemoglobin. As you'll note, they're very different from each other. Let's look at what this is. Percent saturation. That is, what percent of the molecules are full of oxygen? This is the concentration of oxygen. This is measured in pressure. Okay? Now, myoglobin, basically it has what we call a hyperbolic curve. It binds oxygen, and that's it. It has a very high affinity for oxygen, meaning that at very low oxygen concentrations, it's at least 50 to 90% occupied with oxygen. The reason we don't use myoglobin to transport oxygen through our body is because it's good at grabbing it, but it's not so good at letting it go. It only lets it go when the oxygen concentration gets really low in our muscles. There's the battery part. Question?
Why is it a curve at all?
Why is it what?
Curve at all, because there's only one that binds to myoglobin, right?
There's only one that's binding to myoglobin. That's right. And the fact that myoglobin has a high affinity. Okay? So each one is looking to grab a hold of oxygen. It doesn't have T states, doesn't have R states. In order to have T and R states, we have to have multiple subunits. There's a very important consideration. Now, when we look at hemoglobin, hemoglobin has some really cool properties, folks. Okay? Let's think about this. When I am in the lungs, these two curves are way up over here. They join. Meaning that under high oxygen concentrations, hemoglobin's full of oxygen, just like myoglobin is. As hemoglobin leaves the lungs, it goes out to the tissues where the concentration of oxygen is lower. And as it gets into lower and lower oxygen concentration, look, hemoglobin starts giving up oxygen. Hemoglobin meets the needs of our body in an amazing way. Where the oxygen concentration is high, it grabs it, and where the oxygen concentration is low and it's needed, it gives it up. That's really cool. If we were to describe things, we would say down here we are in the T state, and up here we are in the R state. Myoglobin doesn't have T and R states because it's only got one subunit. It can't flip. Because remember, with hemoglobin, one subunit interacted with another subunit, and that's what caused that change. In the case of myoglobin, there's only one subunit. There's only one oxygen being bound. Is that a question?
Yeah, when the hemoglobin leaves the lungs and enters the bloodstream where there's less oxygen, what pulls the oxygen away? Is that a function of it letting go?
Oh, good question. So when hemoglobin lets go, it goes out in the lungs. What lets go of the oxygen? This is like the other things I've talked about here. Remember that when a protein binds something, it's not a covalent bond. Okay? As long as it's not a covalent bond, that means there's an interaction, and it can come off, it can come on, it can come off. So if I'm a hemoglobin and I've got an oxygen, a certain percentage of the time it's bound to oxygen, a certain percentage of the time it lets go and grabs a hold, right? So if the oxygen concentration is low, it's going to be more likely that when it comes off, it's not going to make it back on. The cell's going to grab it. Okay? So that's the reason that happens. Now, there's other things about hemoglobin that are even cooler than this, that tell the hemoglobin where to dump the oxygen. Okay? We can see, okay, the oxygen concentration is going to influence where it lets go. But the body has other signals that tell the hemoglobin where to let go of the oxygen. Let's talk about it. Okay? The first of these I'll talk about is the Bohr effect. And no, that's not B-O-R-E. This is B-O-H-R. It's named for the scientist who discovered it. And it's kind of a cool process. What Bohr discovered was that hemoglobin, in the presence of protons, tends to let go of oxygen more than if there's no protons. Now, based on what you've learned about protons and pH and charge, what would this tell you? There's a change in structure, right? When we put protons on, we're going to change charge a little bit, and changes in charge may have some subtle changes in structure, and you've already seen how some subtle changes in structure can have some very big effects on oxygen binding. More protons means more oxygen is released. And that's happening because we're changing some charges inside of hemoglobin because those side chains are grabbing protons. Well, where do we see increases in proton concentration? In actively respiring tissue. When I'm doing this with my muscles, they are actively respiring, they are releasing protons, and guess what? Here's a flag that says, I need oxygen. The protons bind, and at the exact place where the protons were, oxygen is released. Hemoglobin is giving oxygen where it's needed. Now, that's not all. But wait, there's more. I feel like I'm a late-night TV commercial giver. Okay? I get to say that a lot with hemoglobin. Okay? But there is more. One of the byproducts of respiration, of actively respiring tissue, is carbon dioxide. Not carbon monoxide, but carbon dioxide. Hemoglobin grabs a hold of it. So when it gets to the tissues, it senses the protons, it lets go of some oxygen, and it grabs the products which include carbon dioxide. Now I want to emphasize, carbon dioxide does not bind to the heme group. It's binding to a different part of the hemoglobin molecule. Only carbon monoxide binds to the heme group. But nonetheless, hemoglobin is carrying away some of that carbon dioxide that's there. That's important because carbon dioxide is toxic and we want to get it out of our system. And you thought that was all? There's still more. Here's the graph of what I just told you. Same thing before. What percent are full of oxygen? What's the concentration of oxygen? This is graphically showing you what I just told you in words. There's myoglobin for comparison. Here's hemoglobin. At about pH 7.4 is about what it is in our bloodstream. This light blue line shows us right here. We see that at very high oxygen concentrations, they're all about the same. But if we compare the pH differences of these guys, what do we see? The lower the pH goes, the more oxygen is released, meaning that the lower this curve is. The higher the pH goes, the less oxygen is released. Okay? All right. So that's what the Bohr effect is telling us. Okay? When we look at what the concentration is of the oxygen in the veins, we see it over here. These are lower, meaning more oxygen is given up. We look at the concentration of oxygen in the arteries. There's not so much given up because the need isn't great there yet. All right. I said there's more, and there's more. And this is one of the cooler ones. Oop, wrong one. Well, actually, no, I didn't show you that there. Okay, sorry. There's a reason I put that figure there is to remind me of something. Now, there's the structure of hemoglobin I showed you just a bit ago. Hemoglobin has 4 subunits. And if you look at the structure of this guy, it makes a very neat donut. Right? Donut holes aren't good for anything, right? Wrong. It turns out donut hole of hemoglobin is the most important structure in it, except for perhaps the heme. Why? Well, it turns out that this donut hole is exactly the right size to bind a really interesting molecule. The molecule is called 2,3-BPG. It's got a longer name. You don't need to know it. 2,3-BPG. It's the exact dimensions to bind to that molecule, and that molecule fits in here really nicely. Let's talk about that molecule first. First of all, where is it produced? Well, 2,3-BPG is a byproduct of actively respiring tissue, just like protons. So protons are one signal. Another signal of actively respiring tissue is 2,3-BPG. When it binds to hemoglobin, it forces hemoglobin into the T state. We talked about the T state. We said it didn't want to bind oxygen, so guess what's going to happen if it's got oxygen bound to it? It's going to let it go. It's going to let it go. So not only are protons telling hemoglobin where to dump oxygen, telling hemoglobin where the active respiration is going on, but so too is 2,3-BPG. What happens when these things that are making hemoglobin let go of oxygen get back to the lungs? If we have protons, if we have 2,3-BPG, hemoglobin is not going to want to grab oxygen, right? We're going to have trouble. So there must be something that's working in our favor, and there is. A, the concentration of protons in the lungs are higher than it is in the tissues. Protons get gobbled up. Protons are taken care of very easily. What about 2,3-BPG? Well, 2,3-BPG is kind of like that oxygen. It goes on, it comes off. It goes on, it comes off. It goes on, it comes off, right? The actively respiring tissues produce that 2,3-BPG, but then the hemoglobin leaves those actively respiring tissues, so it's got a long way to travel back to the lungs. And during that trip back to the lungs, it comes on, it goes off, it comes on, and a cell grabs it. 2,3-BPG can be used by cells to make energy. Now, this is cool because it means that by the time the hemoglobin makes it back to the lungs, it's no longer bound to 2,3-BPG. If it were, it would remain stuck in the T state. Putting oxygen on wouldn't make any difference. It would stay stuck in the T state. But when the 2,3-BPG is gone, hemoglobin can flip from the T to the R when it binds to an oxygen. Now, this is really cool And there's one really important health consideration that you should know about this. Okay? 2,3-BPG is much higher in the blood of smokers. Let's think about that on and off and on and off. If you're not a smoker, 2,3-BPG comes off and the cells grab it, right? Because there's not a lot of 2,3-BPG to grab. But what if the 2,3-BPG concentration is already high and you're a smoker? It comes off, another one comes in. As a result, it's much more likely that the hemoglobin of a smoker makes it back to the lung attached to 2,3-BPG. And the reason that smokers run out of breath when they're climbing stairs, you can start to see where this is headed, is that they have a higher percentage of their hemoglobin that's completely stuck in the T state because of the 2,3-BPG they've got. A good reason not to smoke? Absolutely. Yes? That has nothing to do with the nicotine, no. No. Nicotine is another very big problem that more relates to cancer, okay? But it actually has to do with the way metabolism occurs. And we'll actually talk about this when we talk about metabolism in a little bit, okay? Yes, sir?
So after the 2,3-BPG comes off the hemoglobin has no more oxygen attached to it, so it's still going to be stuck in the T state until it gets...
It's not stuck, but it is in the T state. But it's in a flexible T state, that's right. As opposed to being locked in the T state, which is what it is with the 2,3-BPG. When the 2,3-BPG is there, hemoglobin can't flip out of the T back to the R. Yeah.
Is that every case scenario, or is there any exceptions to that?
Well, I mean, the important thing is that you're going to have many more of those tied up like that. So I won't say it's absolute, but it doesn't have to be absolute to stop us from being able to breathe properly, right? Yeah. How many molecules of CO2 get carried out per hemoglobin? That's a very good question. I don't have an answer for that. There are several per hemoglobin, but I don't know the exact number. Yeah.
CO2 doesn't affect like the T state or the R state?
CO2 actually does slightly affect the T or R, but that gets a little complicated, and so I'm not talking about that here. But it does have a slight effect, yes. Yes, sir.
Does 2,3-BPG affect the CO2 binding?
I'm sorry?
Does 2,3-BPG affect the CO2 binding?
Does 2,3-BPG affect CO2 binding? As far as I know, it does not. That's a good question also. I don't believe it has any effect on CO2 binding. CO2 is binding at a different place and in a different way than oxygen is, so I don't believe there's any effect on CO2. Good questions. You want another reason not to smoke? Besides the fact you can get cancer with it, okay? Another one that relates to oxygen, all right? Smoking introduces a lot of carbon monoxide into your bloodstream. You've already seen what carbon monoxide does, right? So not only are you nailing some of your hemoglobin because it's stuck in the T state, but some of the ones that are in the R state bind carbon monoxide and oxygen can't bind. There's two things that's choking you for oxygen if you're smoking. Okay? So, I have nothing against smokers, don't get me wrong. In fact, I feel sorry for people who are stuck with smoking. But it's a good reason to not smoke, and it's a good reason to, if you are smoking, to quit. Okay?
Doesn't it lower your vitamin C?
There's a lot of things, a lot of effects that smoking has. Yeah, indeed, it does. Smoking introduces reactive oxygen species that we'll talk about later. And reactive oxygen species have many deleterious effects in the body. They include atherosclerosis, where we think about plugging of the arteries. They include likely effects relating to cancer. And reactive oxygen species, as a result of trying to inactivate them, that's one of the reasons that you lose antioxidants like vitamin C, vitamin E, and so forth, is because they get used up in basically breaking down those reactive oxygen species. Other questions? Cool stuff, huh? I told you it was a cool. I told you it was a cool molecule. All right, let's see. What else do I have to say here? Fetal hemoglobin. I haven't even finished about hemoglobin. There's 2,3-BPG if you want to see what it looks like. No, you don't need to memorize that. Okay? And that's more reiteration of the same thing I just said. Now, the last thing about hemoglobin is even more remarkable. But wait, there's more. All right? This is the last one, I promise. All right. When we think about hemoglobin, we realize that as animals, we've got other considerations. We've got to carry our young, or at least we, being the females of our species, have to do that. Okay? Fine and dandy. But those, those organisms, that fetus needs oxygen. And the fetus only has one source of oxygen. It can't absorb it through mom's belly. It's got to get it through mom's bloodstream. Well, if the fetus has a hemoglobin just like mom does, it's probably going to fight a losing battle. It's probably not going to get as much oxygen as it needs. So it turns out the fetus has a slightly different hemoglobin than mom does. It's called fetal hemoglobin. Okay? Fetal hemoglobin, I told you that adult hemoglobin had alpha 2, beta 2. That is 2 identical alpha units and 2 identical beta units. In the fetus, the beta units are replaced by 2 gamma units. So fetus has alpha 2, gamma 2. All right? Now the gammas are very much like the alphas and the betas also, but they're very slightly different. And the slight difference is something that you can relate to. If I were to draw the structure of an alpha 2, gamma 2 on the screen, it would look very much like the alpha 2, beta 2, except for one thing. It would have a very slightly smaller place for 2,3-BPG. The donut hole would be a little smaller. And guess what? 2,3-BPG won't bind. The fetal hemoglobin cannot bind to 2,3-BPG. That means that the fetal hemoglobin spends much more time in the R state than it does in the T state. The R state is the state that likes to bind oxygen, and guess what? The fetus can actually take oxygen away from mom's hemoglobin because it's got a greater affinity for it. That's what you see in this thing right here. Here's the maternal hemoglobin in light blue. Here's the fetal hemoglobin in purple. Up means greater affinity. At the same oxygen concentration, the fetus is much more loaded with oxygen. That's cool. But Kevin, wait. If it doesn't have 2,3-BPG, isn't it going to have trouble letting go of oxygen? What do you think? There's one no. Do you have any yeses? There are no yeses? Thank you. There is. It turns out the fetus doesn't have much of a problem with that.
Why?
Except it does. All right? There is a problem. So there's not a problem in the environment in which the fetus lives. It's a yes and a no answer. Okay? So you're both right. All right? Why do we need what's 2,3-BPG doing in our body? It's telling the body where rapid metabolism is going on. Where is rapid metabolism going on in the fetus? Not very many places. Unless it's kicking mom, it's not doing much exercising. Whereas mom is out running, jogging, climbing stairs, carrying groceries, cooking dinner. Very different oxygen demands. Maybe sitting down and watching the tube where you're not using much. Mom has got to respond to a lot of different environments, be able to deliver it under those conditions. But the fetus is just sitting there pretty much just laying around most of the time. So its oxygen needs are relatively constant. And the fact that the fetal hemoglobin holds on to oxygen quite a bit doesn't really pose too much of a problem for it. It gives up what it needs, and that's basically it. That very first thing we saw where the oxygen concentration got lower, the hemoglobin let go of it, that works for the fetus just fine. Make sense? Now, the common question I get at this point is, well, when does fetal hemoglobin stop being made? It's around birth, usually about in the first year of life. Then the fetal hemoglobin will stop being made, and the adult hemoglobin will start being made. Was that your question? Yep. I read your mind. Yes?
So is it always in the R state, or just mostly in the R state?
It's mostly in the R state. Now, I do have a song that covers all this. You guys like to sing it? Would I? I want to hear you. I can't hear you guys singing. If not, I'll just stop doing these songs because I'd like to hear you singing loudly. This one's a real easy one to sing. It's called Hemoglobin's Moving Around. Oh, isn't it great what proteins can do, especially ones that bind to O2. Hemoglobin's moving around. inside of the lungs. It picks up the bait and changes itself from T to R state. Hemoglobin's moving around. The protoporphyrin system, its iron makes such a scene, arising when an O2 binds, pulling up on histidine. The binding occurs cooperatively thanks to changes quaternary. Hemoglobin's moving around. It exits the lungs, engorged with O2, in search of a working body tissue. Hemoglobin's moving around. The proton concentration is high and has a role. Between the alpha betas, it finds a midazole. That's something else. Empty their loads, the globins decree. We need to bind to 3-BPG. Hemoglobin's moving around. The stage is thus set for grabbing a few cellular dumps of CO2. Hemoglobin's moving around. And then inside the lungs it discovers oxygen and dumps the CO2 off to start all over again. So see how this works? You better expect to have to describe the Bohr effect. Hemoglobin's moving around. All right. Good singing today.